Assalamualaikum viewers, welcome to my channel Genome Study. If you are new on my channel, please subscribe and press the bell icon so you will get the notification of all the new videos. We have started the new series of videos and this is the part 2 video. In the part 1, we have downloaded and installed the VARNA software which is used for 2D visualization of RNA RNA interaction or the interaction of microRNA with our gene of interest. So how that Verna software shows results, and this is a video, and we have already downloaded that software. So the link of that video of downloading the Verna software is present in the description, so you can follow that and see how we have downloaded and installed that software. In this video, we will use the RactIP online tool and VARNA software to visualize the RNA RNA interaction between the microRNA and our gene of interest. The link of that RactIP online tool is also available in the video description, so you can follow that. In that analysis, so we can see like this type of analysis in which our gene of interest and that the interaction of microRNA, that how that microRNA interact with our gene of interest and regulate it. That red line shows that the interaction of microRNA with our gene of interest. How we get these results? Let's start. This is the list of my microRNA and that gene. I am working on microRNA 397C, which is responsible for the genes that regulate or responsible for the drought resistant or drought tolerant gene in glycine max soybean. And this is my gene of interest, which is involved in the drought stress, and this is my gene that regulate this gene. So how we get these results, that which RNA interact with which gene? So link of that video that we use, that we have extracted these data of microRNA and our gene of interest interaction is you can find here. This is the link of that video in which we have described this whole process, that how we get these microRNA and our gene of interest and their interaction. So and the link is also available in the video description. So in microRNA or RNA, we need the uracil instead of thymine. So first we need to replace that T into U. So here in text file, go to edit and then replace, and here find what, write U and replace with, find what is T and replace with U, replace all. So these both sequence are that our microRNA and that our gene of interest comes in the RNA shape. Now we go to the Google and write RactIP and then open this link, that RTIPS, that DNA, that bio, that KEIO.ac.jp, that KU University. RactIP KU University, open it, and here, and this is our window. So here you can select, here this is RactIP, and here is your first RNA sequence, that is your microRNA. So copy it and paste, and then you can copy your gene RNA and copy in the second window, second box, In first box your RNA, and then second box your second RNA or your gene of interest, and then click on predict. So now it is computing. So here are our results. These are the interaction results. These are our microRNA, and this is our gene of interest. It shows their alignment or interaction, and this is the energy level that used during the interaction of these two microRNAs, these two RNAs. This is the DL sequence of one microRNA is 21 nucleotides, while the sequence proves that our gene is 759 nucleotides. And also you can get some other results that the details or the interpretation of the output these results. When you click on it, so it will give you these results or some parameter that what they stand for. So if you are interested, so you can read it and then interpret your results from here. And also this is the how to say, this website was made by that Graduate School of Medicine, Osaka, University, Japan, and also Department of Biosciences and Informatics, Keio. So now we need this BPSeq file to visualize and edit our result in the VARNA software. So right click, show in the folder. So first we need to open that our VARNA software that we have downloaded and installed in the last video, or the part 1 of this video. So this is not our how to say interaction model. This is from the website. So we need to open our that file, BPSeq data file that we have downloaded, this one, and open it in that VARNA software. So here you can, in this box, right click, then open, and then find your file where you have present. My file, I have keep that file in downloads, and this is your BPSeq. This is, so it is my file. So here are some parameters that you can use and make it some brighter. Like here, go to view and you can zoom it from here. So you can zoom it by seeing your data. So this, first you need to see that where is our microRNA. Our microRNA is first sequence, so it is present at the start of that this sequence. So we will go at the start of that sequence. Keep the cursor here, press, and then press the arrow, the right arrow, to go to the start of the sequence. So here, this is our 21 nucleotide sequence of our microRNA, and then our gene of interest is started. CAUUGAUG. So here, so now go to the view, and then if you give some rotation, that at which angle we view right click and then rotation, so you can rotate it where you need. So then here you can check that rescale, so smaller or bigger that your base pairs, so you can adjust it accordingly. And then you can also check the border size, the length and width of the border, where you want to adjust it. If you here we can, so this is okay. And then go to the RNA style is toggle means there is circle outside the writing, that if we zoom it then you can easily understand. Okay, yeah, now there is no circle outside the base pair, so here we can toggle so it is circulated. And then base pair style, that here interaction, which one lines are like this, these are lines, or circles between these interactions, between two base pair interaction, or line and hole at the center, or whatever you like, so you can adjust it. And then here show the non-conical base pairs, like if there are conical or non-conical base pairs in your sequence, so it will show or not show, so you can select according to your choice. And here is a customized color to any, different options, you can modify it. Or here color, what color you want, or your base pairs or something else. So here is that is the backbone color of that the structure. This is the backbone that is joined to bases. So what color you want, is blue or anyone, red or anyone, so select it, and then this is changing to the red one. And other one is the if backbone is raw or not raw. If untouched, then the backbone is disappear. Draw. Then you can go to miscellaneous setting, that number period means that these numbers comes after how much, shows after how much number, means that after every 10, like here 10, this is at 30, or after 33 or 14 or 20, like 18 interval. So 10 is okay, I think. Like here you can see is 20, 30, this is a position of that base pair. 40, like this. And other one is the background color, background of this graph color. You can change from here, like like this. So you can change the background of that graph, and here you can also give the title, their font and color. You can choose by your own, and here are some bases that you can see that at one position 2, 3, 4, and this is the color of the base, outer line like this. This is color. You can change it according to your choice. Like here I can change this one, so it is the first base color is changed, or I want to keep it as it is. So from here the name of that, so you can also change the name if you want to change, so you can. Here this is first base pairs and color is changed. If likewise you can change by everyone or anyone you want, so you can change its color, and also the number that here. In this way you can also change that bases color and base pairs. Like here these are the base pairs, UC, UA, like this. You can also change that their color or like style or everything. This is a redraw, that algorithm, that this structure is in linear position. If you want to see that it is come in this way in linear, it's a linear structure, and this is the circle, anyone that you need, or it's a radiant that we are just showing, and I think it is very beautiful to use it, or nav view. So this is the nav view of that your structure, whatever you want to see or check this nav view. And so what, in which style you want to take that structure, in which you think that it is beautiful and it is more convenient or easy to understand, so anyone is that you can take. So then here is the flat or that rectangular. So here is the space between bases, like space is smaller between two bases. If you do this, then it will become bigger, the size of each base, or space is small, greater than the size of each base is smaller. So it's up to you in which you want to keep it or adjust it. And then it's a color by base or by base pairs or by position, whatever you want to give. From here you can change it. Now the important thing is annotation, then new, and then go to region. Then you click on it, so it will show that in which region you want to highlight. So I want to highlight my microRNA, so that start from base pair 1 to 21. So from here I say it's 1, it is 1 because it is start from the 1 just, or you can also see here this is base pair 1, so it will start from here, and then it says 21. So here is 20, this base pair is 20 and this is 21. So we will minimize until it becomes at 21. You can see this one. Now it is 21, 22, 23, so it is 21. So you can drag this to make it up to your position in which you want to see. So here is outline color, this color. So if you want to change it into blue or any color, so you can change. And then this is a fill color, the inside that fill color. So I want this here, this one. So, and the radius, that is the thickness of that is you can also change it from, I change it 8 or any one you can change it. So it will show your RNA. So my RNA starts from here, so it will interact with here is this base of RNA interact with this gene or gene of interest base pair, and then here in this loop it will interact. So you can see one by one here. So in this way you can check your RNA interaction. So we can zoom in. So we can zoom in that customize or okay. So you when adjust it, if I zoom in, and this will clearly show that how our gene of interest or interact with our microRNA, and then it regulate it. Now you can save it by right click, then save as, or no sorry, so export in the form of JPG or any other format, JPG. So in your destination, so RNA interaction, save. Here you can adjust the resolution. I set 600 and here is quality 100% and okay. Then you can check it in your destination. Here is our file, JPG dot so you can open it. So here it is, this is our structure. So in this way you can modify your structure or make your gene of interest with interaction with your microRNA, which regulates your gene of interest. So hopefully this video helps you. If you have any query, so you can contact with us. I will try to solve it. Thank you.